And so thanks, Elena, for the introduction. It's a nice opportunity to present this online. See familiar names that I'm used to see from the industry, also from academia. So welcome, everybody. This is an overview, a bit technical, but mostly to give you the big lines of what we can do with diffusion MRI, AI to assess white matter and start indirectly quantifying inflammation, axonal loss, and demyelination. So like Elena said, if you have any questions, write them in the chat. JC and Elena will compile them, and then we can have a discussion at the end. So the agenda today is to go through a brief introduction to diffusion MRI, the basic concepts that you need to understand to see how we can extract measures of microstructure sensitive to inflammation, axonal loss, and demyelination. And as an illustration of all these, I'm going to use Alzheimer's disease as an example, AD, and the ADNI database, which is a public database with hundreds of subjects, to illustrate some of these measurements in the ADNI database. But keep in mind that we have lots of results also on preclinical data, on mouse and rat models, on multiple sclerosis and Parkinson's. So if you're ever interested in more information, let us know, and we'll be happy to share some info on these other diseases. So let's start. So how do we image white matter microstructure with diffusion MRI? And so before I start, I have to bring some concepts in that we will need all through the talk. Is that basically white matter is a very complex microstructure environment. So at the microstructure level, that is a couple microns big, we have axons surrounded by their myelin sheath. You have lots of crossing fibers. So each fiber population has this little environment of microglia, astrocytes supporting the health of the fiber population and the myelin sheath. And of course, you can throw in there some CSF from partial volume if we're talking about MRI imaging, and of course pathology. And the holy grail really of white matter imaging would be to come up with biomarkers. If I take this little drawing here and I'd extracted just the healthy white matter part of the previous slide, the holy grail would be to find markers that are sensitive to pathology. Like neuroinflammation, where you can see here an increase in microglia activity, an increase in extracellular space. So for today, this is our view of white matter neuroinflammation. Demyelination also, if you start losing some myelin sheath, this will affect the way the water molecule can traverse and navigate in this environment. And so we'd like to find a marker of demyelination, and axonal death or axonal degeneration, where if you start losing axons, you would like a measure as well to be able to quantify this indirectly without using microscopy or very invasive techniques that need markers. And so this is the holy grail, and that we'd like to do this non-invasively with MRI-based measurements. The other thing to keep in mind about white matter is that there's also a macroscopic organization in the white matter. That is, axons tend to organize in tracks or fascicle bundles that I will use in this talk. And these tracks make long-range connection between different areas of the brain. And so it's really important, if we want to assess white matter properly, to have both macro and micro. That is, we know which track we want to study, and then zoom inside to study its microstructure. And just as a fun fact, it's estimated that we have around 160,000 kilometers of white matter tracks inside our white matter in the human brain. So this is a crazy amount of potential fibers crisscrossing and intersecting with one another, which will make the problem even more challenging. And so diffusion MRI is the way to do this non-invasively. Amongst other things, it's very sensitive to the water molecule diffusing in this environment. Here in A and B, you can appreciate some little random walk simulation where you see particles navigating inside axons, in between axons, here in another view with these cylinder views. And so diffusion MRI is a specific MR modality that's able to capture this information. A little bit, I like to compare it a bit like a Google car driving around town, taking tons of snapshots, and then giving this to the Google engineers to reconstruct the street view in 3D. Well, diffusion MRI does this for us. It will acquire different diffusion-weighted images in different directions, taking little snapshots on how the water molecule navigates in these complex microstructure environment. And basically, the water molecule leaves a signature in these images. And then it's a matter of image processing and image reconstruction to access the information about how free the water is, how trapped it is, and try to come up with different measurements that are sensitive to the environments I was just showing you in the previous slides. And just as an illustration, just so that everybody's on the same map, one of the most famous technology to acquire such images is diffusion tensor imaging, DTI, where you do acquire different directions, different images from 6 to 30 or so direction, and you're able at every location of the brain, in 3D, this is called a voxel. So a pixel element is a 2D pixel, a voxel is a 3D voxel, a 3D picture element. And so you're able to reconstruct this. How does the water molecule diffuse in this voxel? And this is very popular, 10,000 plus citation. It's very fast. You're able to do this on pretty much any clinical scanner in 3 minutes. The only problem here with Tensor is the modeling assumption that you can only have a single fiber population in the voxel. And I'll show you today that you can do better processing with pretty much the same data to extract a lot more sensitive information. And so with this fractional anisotropy, FA will quantify how elongated this tensor is. If it's a needle, you get an FA of 1. If it's a sphere, you get 0. And you see, it's a very nice way to light up the white matter. It's very sensitive. As soon as you have disease or pathology, typically FA will drop. You get lower FA and very sensitive to lesions, to anomalies. So very easy to pick up things with this fractional anisotropy. And this is where you have also a reason why you have thousands of papers basically using FA as a proxy of white matter integrity. However, like we said before, the environment can be a lot more complex than a single fiber population. You can have crossing fiber population. Each population has its own environment. You can have pathology, contamination with CSF. And essentially, the tensor model will take just an average of everything going on there. And so even though it's sensitive, it's highly unspecific. And here's just another illustration of this where you have 4 configurations here. In the first configuration, this would be healthy bundle of axons, very oriented and healthy. Here you would have 2 healthy crossing fiber populations. Here you would have a demyelination scenario where suddenly the myelin sheath is deteriorating, so it's easier for the water molecule to diffuse perpendicularly. So you see these tensors fatter than the healthy tensors. And in this case would be an inflammation case where you have an increase in extracellular water. So you have these little pockets of isotropic free water appearing in the voxel. And in all these cases, unfortunately, DTI can more or less give you the same average diffusion tensor with an FA of about 0.4 or 0.3. And so even though it's, like I said, sensitive, it's highly unspecific. We need to do better than this. And we can. So the first problem we need to solve is the crossing fiber problem. Here I'm zooming into a coronal slice of the brain, a big traffic jam of fiber crossings between the corpus callosum in red. In blue, you have the cortical spinal tract. In green, you have the arcuate fasciculus coming out at you. And you can see this beautiful complexity that nowadays we can pick up with proper fiber crossing reconstruction. With similar DTI-like data, we can do this and get the complex crossing fiber architecture. So that's the first thing we need to solve, and that's been solved in the recent years, is estimating crossing fibers. And it's an important problem. It's everywhere, in between 2/3 to 90% of voxel. Somebody just came in and could mute his microphone, maybe. Okay, thank you. The other important thing is, and this is a nice illustration here, imagine you have two groups, a control group and a patient group, and suddenly you see that in your patient group, your tensor seems to be more elongated. That means essentially that FA is increasing with pathology, with neurodegeneration. And so this is a nightmare for scientists and doctors, like, what? What's happening? Why is my FA increasing? Well, this is just another illustration that maybe what's happening is that underneath, you're in a complex three-way fiber crossing, and it's only the green fiber population that's actually degenerating. And so only one of the three fiber population is going wrong. And so in fact, the tensor looks more elongated because you lost perpendicular anisotropy. And so it looks like FA is increasing, but in fact, it's just one of the three fiber population decreasing. Just another argument to say it's critical to go towards white matter assessment that's crossing fiber robust and that will assess white matter on specific tracks. And here's a new term for you. I talked about pixel earlier and voxel for volume element. Now we have this new term called the fixel, which is a fiber element. And so within an imaging voxel, you can have multiple crossing fiber population, and a fixel is just one of these fiber population. And each of these fixel can have its own little microstructure environment, its own axonal packing density, its own myelin microstructure, its own inflammation index or marker, different from the yellow or the orange fixel. And so once you've solved the crossing fiber problem, then it's important to start assessing white matter microstructure along specific white matter tracks. And so essentially now, if I go back to my little drawings, with today's methods, we can solve these problems and use, in the first two cases, proper fiber crossing assessments and do better fiber tracking, and use fiber crossings and also better white matter reconstruction to start accessing these microstructure abnormalities like demyelination and inflammation. And I'll get into that just now. And so how do we do this? How do we use diffusion MRI to get access to these indirect markers of inflammation, axonal loss, and demyelination? And so the first thing to acknowledge is that if you have a healthy white matter axons in a voxel, well, you have a tissue diffusion tensor, and you may have a little bit of extra cellular water lying around. And in case of inflammation or neuroinflammation, you see an increase in extracellular space. This has been really well shown in several papers. And what's happening actually is that there's an increase of free water in your voxel. And so with a little bit more sophisticated processing and reconstruction, you can actually capture these different compartments. The tissue part for the axonal part, and the free water part for the inflammation part. And so for us, in the rest of these talks, this is our indirect marker of neuroinflammation. How much free water is there in every voxel? So this is a number. You can create now images or maps of free water index. The other measurement related to axonal degeneration is apparent fiber density. You see this little drawing here. The more axons you have packed in a voxel, the higher this amplitude will be, basically because the larger the cross-sectional of green stuff it is here, and the less blue stuff in the cross-section. And so again, if you have a loss of axon, you're losing axon density, you're losing some of these little green circles, you're increasing some of the blue surface area, and so suddenly this amplitude reduces, and you can quantify this and link this to axonal degeneration. A little bit like fractional anisotropy, but really specific to the tissue part of the compartment of the voxel. So again, for the rest of the talk, this apparent fiber density marker will be used as our indirect measurement of axonal degeneration. And finally, once you've accounted for crossing fibers, free water contamination, if you're in a case of demyelination, you're losing the myelin sheath, it's easier for the water molecule to diffuse perpendicularly here. And what you see once you've accounted for the extracellular free water phase is that this diffusion tensor part that's really attributed to the tissue compartment, if it gets fatter, you can basically link demyelination to this radial diffusivity of only this tissue part diffusion tensor. So we get our free water, our apparent fiber density, and our radial tissue diffusivity that's related to neuroinflammation, axonal degeneration, and demyelination. And of course, this is indirect. This is using diffusion. This is more sensitive, but of course has some measurement errors. And so now it's also an important challenge to validate this, to quantify this, and to make sure we can do this robustly. And then the next thing to glue all this thing together is tractography and tractometry, to have track-specific assessment of our white matter. What we're going to do is whole brain tractography and reconstruct millions of tracks or fibers or streamlines in our world. And from these whole brain tractography, we're going to start extracting individual fiber bundles that are related to anatomy and to function, such as, let's say, the cingulum bundle that's implicated in the limbic system, the fornix here that's implicated in the memory system, different association pathways that are connecting temporal lobe to frontal lobe to occipital lobe, and as such. So we have now automatic tools to slice and dice tractography now in different fiber tracks. So this, I call this virtual dissection. And so we have more or less 30 white matter tracks that we can automatically extract robustly without any manual intervention, manual placing of boxes and whatnot, and what we used to do in the past, to define fancy regions of interest that really specifically dissect the white matter in different regions. So we can start quantifying what's happening inside and along these fiber tracks. So for instance, we can extract some of the bundles or the fiber tracks involved in Alzheimer's disease. So you see the corpus callosum, the cingulum bundle, the fornix that's coming near the hippocampus. And we can extract just the posterior part of the corpus callosum and start studying just specific white matter tracks. We can quantify volume or spatial extent covered by the track. Is this track losing volume or atrophying? Is it losing cortical terminations? And we can also quantify free water, here in this case apparent fiber density, or some of our other demyelination or diffusion tensor measures inside and along the track. And this is tractography, being track specific to quantify anything in them. So in the past, we've merged this with PET, for instance, with functional imaging, with myelin-based contrast. So as soon as you put these maps in the same space, it's now a very powerful technology to assess pretty much any measurement at the endpoints or inside the tracks. To get something like this, so once you have your portfolio of fiber tracks, then you can estimate average healthy profiles in a population, for instance, and see what is this, for instance, in this case, the fractional anisotropy signature of the cingulum bundle. on average and with 2 standard deviation, so that if you have a disease, a patient coming in, which is this red curve here, you can see if the patient is way off the normality along all the track or just in specific regions. And maybe that specific region is actually a region where you have a multiple sclerosis lesion, for instance, or a white matter hyperintensity traversing the fiber tract. So it's becoming really quantifying basically healthiness and non-healthiness of white matter tracts. So let me, for the last 10 minutes, give you an illustration of all these metrics or measures in an Alzheimer's disease application. So just as a reminder, and that's a nice read. If you don't know this paper, it's a nice review paper. It's actually surprising to see how little white matter is present in the Alzheimer's disease literature. In the kind of disease progression of normal controls to MCI to AD, we rarely see a curve showing where does white matter come into this pathology. We see a lot of atrophy of the hippocampus, swelling of the ventricles, but it's thought that white matter anomalies probably precede all these gray matter anomalies. And so it's very important to pick up these white matter changes early if we want to ever have an attempt at producing drugs or technology to detect these patients early. And so here's a nice paper that was just published as a nice partnership between Imeka, Pfizer, the ANI consortium, and some of the students on this paper is also part of my lab, to show you that a very basic free water estimation can actually capture very sensitive differences between normal controls, MCIs, and normal controls in Alzheimer's disease groups. And this is on the ANI database, only a subpart of ANI with the diffusion data, but that's still something around 200 or 300 cases. And so just as a reminder, every voxel of the brain will be modeled as a free water part to capture the free water unrestricted diffusion, and a tissue part that will capture the healthy white matter underneath this extracellular space. So the diffusion tensor will be used to quantify corrected DTI measures, and this is our free water index. And the pipeline looks something like that. I'm not going to zoom in every step, but I think it's a nice excuse for me to bring in a bit of the AI and machine learning going on in my lab and at Imeka, and show you examples of how such tools can affect the pipeline. And so, for instance, you see here lesion segmentation, you see mask processing to combine free water model in just a safe white matter mask far from the CSF to make sure we're not contaminated by free water CSF, so far from the ventricles and far from the convolution of the external brain, to just compute what is the average free water in this mask. And so AI is really helping us first leverage big data, make sure we can process lots of data, do automatic quality assurance to make sure that we don't spend hours just doing QC and QA and reprocessing things. And for us right now, it's really helping us to do brain extraction and lesion segmentation, amongst other things. And just as an illustration, here's what our AI brain extraction tool can do on T1 and T2 weighted images. Really high throughput, a lot of data sets, and on the B0 image of the diffusion weighted images. And for the scientists out there, this is typically the part of the pipeline that always breaks in 10 to 20% of cases. So if you have 1,000 cases, that's 100 to 200 brains that you have to go and manually edit or relaunch or remove from your study. And suddenly with these tools, almost 100% of cases go. Another place where AI will for sure help in the future is extracting, segmenting white matter hyperintensities or MS lesions or anomalies in the brain. And for us, diffusion tractographers, this is critical because these are potential holes for tractography. So it's really important to segment these parts to complement our white matter mask to make sure we're allowed to track through these regions so we can study them with or without them in the statistics. So this was my little parenthesis about AI, but just to show you that AI is becoming more and more important in all of these bricks of the processing pipeline. So back to our paper, I just want to say that it's a very simple method, nothing fancy about this method. We're estimating a safe white matter mask, safe, which means far from the ventricles, far from the CSF, and we're going to take an average free water index in this. That's it. And what we clearly see is a differentiation statistically between the normal controls and the MCI. You see here the demographics of the data from the ANI database. So 71 normal controls, 100 MCIs, 58 ADs. And then we can also differentiate normal controls from Alzheimer's disease very well, very clearly. And interestingly enough, if we look at where are the most important differences and what's driving these significant differences between the groups, you see the Z-scores lighting up in the brain here a bit everywhere. But if you start mapping them on white matter tracks, you see mostly the limbic system lighting up, such as the cingulum bundle, the fornix. And so this needs further investigation. In the paper, we really stopped at a simple, easy method that we can roll out nicely in clinical trials. But we could start doing tractometry of all these data points to see where in which tracks is happening. And I just want to make sure that there was no significant difference between MCI and AD groups, in a sense showing that the level of free water index in the safe white matter mask was already quite higher and similar for both MCI and AD groups that are already, in a sense, affected by the disease. Now, you may wonder, how do these things evolve in time? Can you picture longitudinal changes? In fact, you can. Here's a 2-year progression. So 24 months, where you see in green and yellow, the normal controls and MCI group, pretty stable. And this is across a whole white matter, safe white matter mask, as opposed to the Alzheimer's disease group really increasing in their free water index across 24 months. So this is true in the whole safe white matter, but it's also true in specific white matter bundles, mostly connecting the temporal lobe. And similarly for axonal degeneration, if we look at what's happening with tissue FA or apparent fiber density, we see here that most people with over 24 months have a decrease, and that's worse for Alzheimer's disease patient than normal controls. And the decrease here, the slope is a bit less steep, but still something is detectable and changing over a 2-year period. Again, most bundles affected are connecting the temporal lobe. And lastly, I wanted to show you some correlation with other markers in the ANI database that are non-MRI. One is PET-based, the AV45, and the other one is NFL, the neurofilament light chain. So again, over 2 years here, we're measuring changes. So delta changes and correlation between changes. That is, what are the changes in free water and axonal degeneration linked to changes in other markers available in the ANI database. And the first one is AV45, which binds to beta amyloids, which is thought to be linked to neuroinflammation. So the hypothesis here is that if you have more AV45, you should have more free water index for these patients, or more changes in these free water. And this is exactly what we see in the data. You can see here now the group size, I have to admit, goes down quite a bit because even if it's ANI database, there are not that many subjects with all the data necessary to do this analysis. So that's a patient that have both diffusion and PET for every time point, but you basically see, and here it's a case that it's not happening for everywhere in the white matter. It's happening for specific white matter tracks, that the more AV45 changes you have, the more free water changes you have as well, with a pretty nice correlation. And lastly, NFL, which is the neurofilament light change measurement, which is an invasive measurement in the CSF through blood. The hypothesis here is that if you have loss of axons, axonal degeneration, you should have debris of axon that should accumulate in the CSF, and so it should be leading to an increase in neurofilament. So an opposite correlation here. And so this is the hypothesis. Again, this is what we see, that the more neurofilament you measure, or the least neurofilament you measure, the higher the less neurofilaments you measure, the more apparent fiber density loss, decrease. Again, not in the whole white matter. It's happening in several white matter tracks connecting the temporal lobe. So this is to show you some of the potential of these measures, how they correlate to other more invasive and expensive biomarkers, and the potential they have to maybe detect changes early that do have an effect longitudinally. So in conclusion, I hope I introduced you to diffusion MRI and the potential of diffusion MRI. I highlighted the importance of being specific, so to include more advanced processing and reconstruction tools that are, by the way, usable as of today on DTI-like data, but we just need more sophisticated processing. The importance of being sensitive to crossing fibers and to different fiber tracks and pixels. And this was a perfect illustration of cutting-edge technology bridging tractography and microstructure imaging, but in a pharmaceutical clinical trial setup where we don't necessarily have access to the fancy human connectome projects and fancy scanners that us researchers have. So I hope you enjoyed this. I really want to thank the whole team at Imeka, especially in these special times where everything is happening online and we meet every morning and we have a good time. And with that, I'd be happy to take some questions and just leave you with this slide just to show you that we're present in, Imeka's present in North America, in Sherbrooke, and we have an office in Boston, Cambridge, and we also have a strategic partnership now with MIAC in Switzerland and Basel. And MIAC is a CRO that's been doing clinical trials for the last 25 years, especially in multiple sclerosis, among other things. So again, thanks for showing up. And if you have any questions or you want extra information about preclinical data, Parkinson's or MS, please don't hesitate to ask. Thank you. We can open the floor for discussion.
Yes. So if anyone has any questions, could you please type it in the chat?
I see lots of familiar names. Hello, everybody.
Okay, so there's one question that I see here for Max. How much could your results be driven bias by atrophy of the specific region?
Which results are we talking about? Maybe can you be a bit more specific? But while you type your answer, I have to say that this is why in the Pfizer paper, we had to define a safe white matter mask to be as far as possible from atrophy and increase CSF. Because of course, CSF is also free water. So if you look at my little diagram, free water can come from the pathology and the increased cellular space, but also from the CSF part. Luckily, they most likely have different diffusivities, so we can try to tease them apart. But the easiest way to tease them apart is to make sure you're far from CSF contamination. And so that's the way to be as robust as we can to atrophy and ventricle swelling.
Okay. The example given is the limbic. Can you be specific about that?
In the limbic system, but so yeah, similar answer again. But this is where also fiber tracking helps. Essentially, if you do reconstruct a coherent fiber track, that means you're under white matter. And if you're under white matter, yes, you can be kind of contaminated by partial volume around it. But this is why it's essential to take it out of the signal to make sure that your tissue quantification is as free as possible from the contamination from partial volume.
Okay, great.
But nothing is perfect, as you know, especially with MRI and the limited resolution.
Okay, great. Does anyone have any other questions? If you could please type them in the chat, that would be great.
Don't be shy. I put on a shirt today. Use me as much as you want.
Any questions? Any questions about the neuroinflammation component or the longitudinal data that we showed?
The AI, our AI expert is on the line, on the line if you want. Yeah, I see the question.
Okay, if you can see the question.
So we have a question from Mohammed. Hello, Mohammed. How is it going with the development of lesion segmentation? It's going well. We have to admit one of the bottlenecks for us developing AI tools is the low number of high quality annotated data. So this is clearly a challenge. So in the future, we hope that we can start working even more closely with MIAC, our new partner. They have years of experience with MS and lesions, with manually annotated lesions with radiologists. So I hope we can pool all this data together and have a better lesion segmentation tool in the future. For now, I have to admit, our lesion segmentation is truly based not to be very pixel perfect, but to complement our masks for tractography. The real big issue for tractography is to have holes in our masks. And so even if our lesion segmentation is not pixel perfect and even maybe a bit too permissive, we combine it with our white matter mask to make sure we have a full white matter mask for better tractography and better track assessment. But I can give you another example is that, for instance, our AI brain extraction tool really started being incredibly good as soon as we hit the 500 subjects to 1,000 subjects in the training data. And with brain extraction tool, it was easier in a sense to correct and to bring the training data higher because we could use automatic techniques like ANS and FSL, and then our employee at Imeka was manually checking which ones did work well and not work well and just boosting and boosting and boosting the database. And at some point, even with a very simplistic, in a sense, deep learning architecture, it was quite feasible to get very good results. So we're hoping that something similar will happen with lesion segmentation as we increase the power of our databases.
Okay.
There's a question from Gal. Thanks for the talk. The comparison with PET marker AV45 was interesting, but it is not a specific marker of neuroinflammation. Of course, yes. Do you plan to repeat the same analysis with TSPO? Yes, of course. Not of course, but it's something we have in the books right now. It's I don't know what I can say or not say, but maybe Elena, you can help me here.
Yeah.
But we do have an ongoing project trying to compare diffusion free water with TSPO.
Preclinically.
preclinical models.
So we should have that data, hopefully. I mean, with COVID, we've been pushed back a bit, but we plan to have it in the summer of this year.
Yeah. And also, there's a few papers out there trying to validate free water with spectroscopy. Not full papers, but abstracts that we could share with you, Gail. And another question from Mohammed is with the data heterogeneous. And yes, that's one of the big challenges, this heterogeneity of the data and MS data or pathology data. So that's why you need big numbers and lots of data.
Are there any more questions that anyone has? Did we answer the TSBO question okay? Okay.
Yeah.
Excuse me, I would have a question. It's just that I can't type in the chat. Could you hear me?
Fine.
Yes,
yeah,
go ahead. Fine. We hear you, Valerio.
All right, thank you. So I have a little bit of a provocative question, actually. So how do you think you can really increase the specificity of your novel diffusion-based marker without having, a ground truth to compare with? So in other words, your patient are labeled, and this label is based mostly on behavioral data, like this patient would have Alzheimer's disease or other pathologies, which is very heterogeneous anyway. So it's not just that all D patients, they might follow the same trajectory. So my question is more like, do you see potential in preclinical MRI, so where you have a more well-controlled condition to bridge this gap of knowledge and to explore more the specificity of these markers? And I was wondering if you are working also in that direction. Thank you.
Yes, for sure. Thanks, Valerio. Due to time restriction today, we decided to focus on, well, first an introduction to the technology and a bit of results on AD, but we do have a whole slide deck on preclinical data. And it's important to us. It's important to me as a researcher, but also for us as Imeka. We have lots of contracts and contacts with preclinical CROs, trying to validate this to have, as you said, very controlled models and that we can follow in time, and that we can also sacrifice and do the microscopy, the histology, and the different markers of microglia activity, myelin staining, and whatnot. And so not on Alzheimer's disease, but on focal lesion and MS preclinical models, we have pretty striking and fascinating results showing the potential of this technology, showing that, yes, we can still be specific and we can be robust to this water, basically, or this inflammation part that can really throw off basically any MRI acquisition, whether it's myelin specific or just tissue specific or anatomy specific. As soon as you have a very high amount of edema and water, you basically are clouded. Your view is clouded, and you see nothing underneath, even though there's lots of things still underneath. And you see this as time go by and edema disappears, that the tissue part is still there. But I agree with you. When we go to these ADNI database and this Pfizer paper, the groups are so heterogeneous, and you see the cloud of I'm not hiding the data from you. The variability is huge. The points cloud is spread. So there's probably lots of different subgroups even in these groups. And so I think the hope for us in the future is we may start using also tractography and track profiling to do better subtyping of groups, so that we have a better chance to just study the people that are, that for instance, have an inflammation problem and not a vascular problem, or axonal degeneration problem versus a demyelination problem. Because at the moment, all these people can be in the same group. They all show a reduction of FA, but they have lots of different concurrent pathology happening.
Yeah,
so it's
absolutely.
just the beginning, to be honest. It's really exciting, but it's just the beginning.
And if I may, do you see also potential for deep learning for, you know, bridging this scale? So where you have,
Sure.
you know, you train a model on a preclinical, and then you explore the clinical side.
Okay, that's a good point. I wasn't thinking about that, but yes, I think, and in some literature in diffusion, we've seen this in the past where even people use simulations. We're getting pretty good at simulating these little environments. So you can use both simulations, so pure synthetic data where ground truth is clear-cut, you always know, preclinical data, and then the bridge to human data sometimes is difficult, but I've seen, you know, successes in that, especially simulating the microstructure environment and trying to bridge also the resolution gap, because that's also a big challenge, because you're really in preclinical data or in simulation, you're actually measuring at the micron level, but in human MRI, you're measuring at, you know, the millimeter level or tens of millimeters, which is, you know, a factor of 6 resolution gap. And so that's a huge challenge for the future, for sure.
Are there any more questions? Does anyone have any more questions about the technology, what our capabilities are at Imeka, what we could do? If not, we can wrap this up. So again, my name is Helena Mystery, and I'm the Director of Business Development for Imeka, located here in Cambridge. We'll send out a follow-up email hoping to get feedback from all of you. We would love to host more if this is of interest to you, and feel free to shoot us an email and ask any questions, and we'd love to talk with you more.
Yeah, thanks a lot, everyone. Stay safe in Europe and everywhere, and fingers crossed for the summer to come in Canada.
Yes. All right, thanks everyone. Have a great day.
You.
Thank you.
Bye-bye.
Bye now.